Today we're going to be teaching you about DNA replication, really exciting process. So, first off, we have helicase. I don't know if you can see this, you might want to come forward with the camera. The helicase is replicated by this red lollipop right here, and it is...
Represented.
What?
You said it's represented by this lollipop.
That's coming up, and it's splitting a strand of DNA in two. And it's followed...
It's breaking hydrogen bonds.
You want to start off from here? You want to keep going? This is primase, and it is following the helicase up and is laying down a primer for RNA to latch on. For RNA, where is it? RNA polymerase to come on and it to attach, and it attaches one nucleotide at a time through hydrogen bonds, such as this. Am I correct? Right, and it connects such. And the blue here is not the thymine it usually would be in DNA. This is RNA, so it's a uracil. And as shown in our key right here, adenine is represented by the red, thymine is represented by the orange, cytosine is represented by the yellow, and guanine is represented by the green. And as previously stated, the uracil is represented by the blue. So this sets it down here, and you got this from here.
Cytosine and guanine have 3 hydrogen bonds represented by 3 toothpicks, and adenine and uracil, or adenine and thymine on this side, would be represented by 2 hydrogen bonds.
Oh, just some confusion here. We realized that we accidentally went from the lagging strand first. The leading strand should have been first. So we're gonna put this into place, and it attaches one nucleotide at a time, up from the bottom. And in our key, as we're just gonna say this so everybody's understanding. So the phosphate bases are the Rice Krispies showing the form. And then the deoxyribose is the marshmallows at the base. That's the sugar. And for the RNA, it's the raisins. And so the hydrogen bonds are blue toothpicks, and covalent bonds are the normal toothpicks. All right? And so...
Okay, so then, this replication, this placement of nucleotides was done by polymerase 3 here, and then now polymerase 1 is coming onto the primers, and then it replaces them with normal DNA nucleotides.
Annoying process. Has to be done. Oh god. Right. So, yes. And so, these are called Ogasaki fragments right here. You can see that on the lagging strand, they are not attached. There is a separation. And so, to make these into one strand, like a complementary strand to this side, you have to take DNA ligase, right? Mm-hmm. Right. And so, sorry, question myself there. This is not chewed gum. This is pink taffy, just for all of you to be sure. And so this comes and it connects the 2 Ogasaki fragments into one long strand.
Oh, so then this replication of DNA is called semi-conservative replication because one side is new and one side's partly conserved or old, and that happens to both sides.
Oh, and also this is eukaryotic DNA replication. Just we did not show this here. This is a more complex form, but there's prokaryotic, but it's just like a zipper going twice, like 2 different ways around a circle, starting at one point and going around the circle till they meet on the other side. Whereas in eukaryotic, they all start in many different places. Is that our presentation, Jakar?